Okay, we will start our next section, and welcome back to the Symposium 34. And in this session, we have 6 talks. The first speaker is Mr. Yu-Heng, Yu-Heng. He is an undergraduate student from the National Taiwan Museum, and he will give us a talk about the significance of citizen science to the Nature History Museum, to Nature History conservation in Taiwan.
Good afternoon, everyone. I'm 20 years old now, and I'm from Taiwan. I'm third year now studying life science at National Taiwan University. So I'm not a researcher, but I do specimen and such as taxonomy, study skin, wet specimens, definition, and so on. So I was a student assistant of Zoology Museum in NTU before. I would like to learn anything about natural history and collecting. So if you ask me what I'm doing, I will tell you that I do nothing but everything. And today, I'm here representing myself and my friends. We would like to share the care cases using issues happened in Taiwan. What's the problem and what we can do and what we want to achieve. So the story was started when I was 6 years old. I started collecting many natural objects at that time. I collect further skulls, sea urchins, skeletons, mushrooms, and everything I saw. So including the fresh carcass. But I didn't know how to do specimens then. I just thought they are beautiful and I don't want them just become rotten and gone. I want to keep them old. Oh, by the way, this is me testing the grass. So when I grew up, I learned how to use internet. I started searching how to make specimens. I just treating them just like beautiful collections. Obviously, I have destroyed many precious carcasses because I'm not a member of those researchers who get data from those dead bodies. I didn't know how much important those carcasses are. This is a little tern I found on the beach, almost dry, but I still following the protocol from Google to cut the body and remove its trunk. If you want to know, it becomes a bad taxidermy. So, until I got in NTU 2 years ago, I finally understand how important those dead life can be for researchers. In the past, I know how hard to collect a dead body, and now I also know how hard to get a useful data for a researcher. I started treating those collections differently. As you see, after a few years, I changed the back taxidermy, become a part specimen, which is providing data and now sent to the research unit. So now I'm using an example to tell you what may happen after an animal dies in Taiwan. One day, a friend messaged me at 11 p.m. when I'm going to sleep. He told me, there's an animal hit by his friends and died in front of the dorm. 3 minutes later, I found the animal. It's a Paguma lavata. When I take up my phone and started shooting photos, the cleaning unit arrived. So I won't find anything if I'm 1 minute here later. In other cases, people may bury it, call for pet undertaker, throw it into trash can, and so on. Or it may roll by cars again and again. We always call that cookies in Chinese. So then I post the data on the Taiwan Roadkill Observation Network website, which collecting dead animals. Collecting dead animal data through citizen science. People can report any single case of dead animal online. And if they have the ability to do, they can send the carcass to the Taiwan Biodiversity Research Institute for free. And this is the most important way that can collect dead vertebrate carcass in Taiwan. And if any researchers need some samples, they can apply the carcass they need, which are good things that citizens helping to collect. Also, only those who took text on species identified can see the location data, so we can also help to collect those bodies normal people doesn't have ability to sense. There are some examples in later presentations provided by Ji-Heng, and everything seems perfect, right? Why do we say there is a problem? It's just another example in many STEM cases. It's a fairy pizza sent to Taiwan rock-killed observation network the same way, a beautiful, completely and fresh body. And there is a laboratory studying paleontology need bones from living animals to compare with fossils. They ask for many completely and fresh body to do bone specimen. They make the specimen in the same place we make our own, so we can chat and exchange our experience, and wow, that's a good thing and the citizen science work. But they said they don't have enough time, money, experiences to do not record morphological measurements, necropsy data, tissue samples, and parasitic evidence collect. That sounds strange and awful, right? So sometimes they even identify the species wrong. This time, we help them check the collecting data using the Google map and see the location the bird's die is surrounded by glass. It means that the bird is more likely from window collection but not rock killed. The person uploading the case was very likely recording it wrong, something worse. They are bad at doing both specimen. They let the new hand doing protected wildlife with suture knife cutting the muscles from the bones. As you see, two tight bones are broken by the new hand. Actually, they don't need that fresh body to do bad bone specimen. So we noticed the problem and we assemble a team. We are students knowledgeable in specimen preservation techniques and data collection methods. We want to establish an improved carcass relay station. Not only collect carcass, but also prepare specimens with comprehensive natural history records, which we used to do it, but we want to do it better. So before we cut in the carcass, we do file notes first. We note the location, times, names, and the other information we note. Then we do morphological measurements following measuring bird published by Chris Media Nature. We check the animal and see if there are any ectoparasites dropped on the clean table. Others, we note everything we notice, such as evidence of injuries, molting patterns, and other observations. Finally, we can open the body. After we cut the carcasses, we can note the necropsy data because we have tried to cooperations with vet, and it turns out as a very pleasant cooperations, and lost data can help to correcting the cause of death. Also, we collect tissue samples. And no endoparasites were left behind. We collect every individual and we note the position we found it. In the family, we open the digestive tract and identify their last stoppers. This information also shows the interactions between species. And it's time for specimen preservation and preparation. We have our guideline system to decide what specimen the carcass can make. The highest one is the skin mount. We can also keep the trunk bone like bone specimens or definitions. The eyes can also keep in ethanol or make it transparent to see the ring. Yeah, actually, we can keep trunk bone and eyes as byproducts always if the carcass has it. Most of the time, We are doing the study skin, and we may cut the wing with the speculars, feathers, and we make it wide open. Sometimes we have the steel carcass or the whole skeleton are needed. We will make the further atlas and upload the image on the further-based website. With the uncompleted or steel carcass, we also made a part specimen, and our idea is that we are trying to keep as much things as possible. Oh, and there is a Facebook group that we post the samples we collected from digestive tract, and many researchers will help us to identify the species in their bodies, and sometimes they will ask the sample from us. Finally, that's why we are here. I'm just here want to learn more experience from you guys and involving in national history better. And the background is those definition specimens I made. That's why I'm crazy about too, and I always want to collect every kind of fish and other vertebrates in transparent. And thank you for listening.
Thank you, Yu-Heng. I have a question for you. In your experience, what's the most challenging and most difficult part in this kind of work?
The most
citizen science.
The most difficult part, I think, is that to collect an animal when I see it on websites, because someone posted it and give us the location, and we arrived. We want to get it, but we couldn't find that. And sometimes
so someone removed that.
And sometimes we just ride motorcycles and 1 hour, 2 hours away and got there nothing.
And how about the funding?
Funding?
On your own cost?
On your cost or
funding?
I'm still, I don't care about that. So, and we do it and we paid our own money to do this. Because we love it.
A great job. So any questions or comments for Yu-Heng? I think the young generation did a great job by their enthusiasts. They love to do that. You? No comments?
No? Bye-bye.
Okay, thank you. Thank you, Yu-Heng.
Thank you for listening.
We still have 1 or 2 minutes, and the next speaker is Chi-Heng Hsieh Chi-Heng. She's also from National Taiwan University, and she has some experience working in the zoological museum in the university. And okay, that's welcome, Chi-Heng. And she's talking about the bird window collection story in Taiwan. Welcome her.
Hi, I'm Chi-Heng Hsieh from Taiwan. I'm in my last year's master studies. As some of you now are from Taiwan, you may know me about bird collection studies, but to be honest, I spend most of my time preparing specimens, just as the previous speaker introduced. So that's why today I'm here not to share about the research part, as we are not bird collection researchers right now here, but the experience in forming a collaboration network in between vets, specimen collectors, and the citizen science providing data. Okay, I think it's a common sense to all of us that these deceased wildlife are precious. We answer all kinds of questions from what we can find on them, including the occurrence record, death cause, species interactions, and so on. However, there has been long, okay, I go the wrong way, okay? So there has been a long, what I so-called the tug of war between the two S's. One is on the vegetarian side, which the vets are telling people to keep away from carcass, be aware of there is a circumstances that they might get disease that are transferred from animals to humans. And in most cases, they will ask to burn everything for safety concern. Another is the collection side, where most of us are right here sitting, and I think most of us would hope to keep something at least for the future. I will say there's no right or wrong to speak for the need of safety or for specimens, but this is causing problem because both sides are all professions. Which way should the citizens listen to, even though they wanted to help and they are not know which way they are going to follow? But is there no solution to this? Of course there is. Otherwise, I'm not going to be here sharing. So we now know that the conflicts usually come from misunderstandings of unfamiliarity. So if someone who knows bits of both could act as a bridge in between and bridge them together, they could as well collaborate. So this is what I was doing for the past 4 years, thankfully to the co-author of this sharing, the open-minded vet serving at the Raptor Research Group of Taiwan. So before changes are done, let's first get through what the traditions are. I think people who saw this photo, I've tried really hard to go through the galleries and trying to look one that is at least good looking, will know that this is not going to make a good taxidermy or specimens anyway. There has been some problems to it when it goes under tradition necropsies, including that for this one, this is a case of a roadkill small Indian civet, which is like a carnivore, so they are looking for whether it is died from rabies in case of safety. So you can see they have removed furs, chunks of furs, to see if there are evidence of injuries. And then the vets doing necropsies, they tend to do large cuts for convenience and also for clearness. For example, this one here has it all the way up from the abdomen to the jaws, to the tip jaws, which is really hard to repair. And as we expected, parts like ribs and organs are sometimes taken away without measurements, and they don't note which pieces are missing. And for through necropsy, they ask for total unfreeze, best for not ever be under 0 degrees to prevent ice crystals from forming if they are doing slices. So, and they are also doing careful examinations, which takes time. Like this one we suspected underwent a necropsy for nearly 6 hours. So then you know that if a carcass is unfrozen and it goes through room temperature for 6 hours, it's just going to rot away. So what could we do to overcome these obstacles? Let's look at them one by one. For feathers and fur removal, we could replace this process with using wet alcohol to wet the furs so the fur lie down and it also expose injuries, or simply ask them to check from the inner side of the skin, because most injuries could be seen on both sides. There are traces to look for them. And for large cuts, we could introduce how specimens are prepared, how we skin them, and what are our needs, to what extent, if we really need a larger cut, what is repairable and what is not. And for the missing parts, this requires the collectors to stay beside to do necropsy with them and make sure the parts are returned or measured before they are taken away or sent away, which is out of reach. And for rotting, it's just simple. What we did is we bring an ice bucket with us to make sure as fast as it could, after the carcass has been dissected and looked through, to get back to a low temperature container to get rid of the insects and mold spores and all sorts of things to prevent it from further rotting. Let's look at the case here. And so this is the first case when I collaborated with the vet at the Raptor Research Group of Taiwan. So you can see that the case note is 001. So this is a crested goshawk suspected to collide on campus, suspected to die of collisions. So what we did first is we go on site and we collect all the field notes we can, and these are also sent to us if a community scientist is doing the same job. So the first thing we do before necropsy starts is always to get all the measurements, including weight and a guess of sex and age, if possible, from the outlook or the traces if we can find it. Then after we start necropsy, we make sure the photos are always taken and that the necropsy waste is done from layer to avoid artifacts and further destroys. So it goes from revealing the muscle layer to reveal the organ layers, and then after checking the organs are removed system by system to reveal the skeletons, and then to places that are in need for further examinations. For example, in this case, because this is off-bit collisions, we wanted to look if there are injuries in the spines or in the brain case. And you can see that probably in the right side, if you have some experience of specimen preparations, this is a way of peeling the skin from birds. So there is no cut, but we just flip the skin through the neck and to expose the brain case. And then this is what we get finally. We will get a wildlife necropsy report with all things recorded and measurements on another sheet, and also doable specimens. So we can see that in this case, after the all necropsy things are done, we can still get a nice study skin and also spread wing, which is just useful for like specimen collections. I would like to pinpoint that an interesting lesson that we may learn from vets for collections. So this is a collaboration, not only that specimens, collectors, museums get benefit, but also vets get benefits. But I will first start like what the benefits get from museums from the vets. You can see that there is a number circled here in red that is called the BCS in short, and the full name is body condition score. I would wonder that any one of you here have heard of body condition score. Any one of you? You could raise your hand. You are from our group, you know body condition score. So we see that most people don't know body condition score because this is a common method for vets only to keep record of whether the animal is in a good shape or not. They have this kind of evaluation scales to see if the animal is ideal shape or is too fat or is too skinny. In contrast, this is a screenshot for the specimen recordings in the avifauna of Taiwan, which are recordings gained from the collections. They illustrate and they try to look for weights, but we see that no weights are recorded, which is interesting. And from what I have personal interviews with the collectors, asking them why don't they record the weights, what they say is that because they are not sure about whether the weights they are recording are correct for the species, as they have no way to evaluate or double check it. There is a more extreme case in this is the scale, I forgot to show it. And then there is a more extreme case here shown in the field guide of wild bird in Taiwan, which is too overconfident. So their previous one is like they have no confidence, so they decided not to record it, but this is too confident, and they said that the weight for the same species I showed before is about 750 grams to like about 1.5 kilograms. And if you still remember the numbers I showed from the previous case 001, it's like 520 grams for a female bird. And if you have some knowledge on raptors, we know that raptors are sexual dimorphism and that females are always larger than males. And this is a female bird already scoring 4 on body condition score. That means that 750 or even like existing 1 kilograms, this is just too much for the species, which means that the numbers of these books recorded are obviously wrong. And in future, if we wanted to correct them, there is a way to do it. Just to go through the collections, if they are not prepared yet, we can use the body condition score to find the ideal shape, and then we have a reference to see if we are recording the right weight for the species. And as I mentioned, not only collectors could get benefits from vets, but vice versa. Museums hold techniques of presenting and preserving stuffs. For example, the diaphonoid specimens, or we know as the stand and clear specimens techniques, where bones and particular tissues get stand, and this is not known to vets. And I just know that not most museums do it, but some museum knows it. So like in this case, we are able to show the minor bone fractures in between T2 and T3. That is where the nerves controlling the wings are, which may not be as straightforward, but this is where a bunch of organs, including the lungs, the hearts, the nerves, all come together beneath, which means that even if they are taking an X-ray picture, the photos are hard to interpret whether there is a bone fracture. Therefore, through the preparation of this clear and stand specimen, we could answer why there are several cases of bird non-collision victims that couldn't stand on their feet or are paralyzed, meaning that they could only move their heads around and they couldn't just mobilize their wings or feet after they died. You may ask that why we are still answering what they happened to them after they have passed away, as this has no help to them. But this knowledge could later refer back to clinical treatments for future patients, meaning that now for vets, knowing that there might be some bone fractures in the spine, in future cases, if they get a bird non-collision case, they can start to treat it beforehand, even though they don't see it from X-rays, giving them a higher chance for the bird or victims to survive. And what's left is that we could put this all up into an exhibitions. And through exhibitions, the citizens who are helping or potentially helping, they could know more what is going on, and that if they are able to bring the carcass in, what will happen to the bodies, which convince them that the bodies will be going under a good care, and they may see them somewhere in museums for educations, which is a proud and most of the time is a motion for people to join and to engage. And is there more potentials? Of course. Later on, we've also developed ways to meet the needs for multi-specimens. And there's a huge amount of data. Because we are doing necropsy and measurements and specimens are also preserved, up till now, like for this, there's 1 year of trial, and then later on we do the same protocol through for 3 years. There are more than 300 birds that have gone through this process, meaning that they all have this full set of data, from measurements to necropsy to specimen, which I think may hold a lot of treasures, which are worthy to, I think, all kinds of natural history museums. And probably this way can also be sent or be interpreted to different museums. And now here, I have still 1 minute, but I'm thanking you for your attention, and we can have further discussion. Thank you.
Okay, thanks, Chi-Heng's great presentation. And anyone who has question? No? No?
Any kinds of questions? Because I know that not many has been working with that.
Okay, I have a question for you because I do feel like it's a really good example of the participation of the citizens, but in the end, they're going to put their hands on the dead body of the animals. So during this activity, who takes the responsibility, or they have to do this on their own risk? Because you have mentioned that is still the possibility they carrying virus or so.
So what we did is we discussed with the vet to what extent that the public could do, for example, at least putting gloves and you are not touching them with bare hands, and you do wash your hands before you are touching other things after you touch the carcass. We discussed with the vet if this works, which I'm not sure if museums tell the people, if people are bringing the specimen, because we see cases when people just take them with bare hands and they just bring it into the museum. And we try to spread this knowledge on exhibitions and also on Facebook pages, on all sorts of cases, to tell them that what should you do if you are going to do this. So this is like a both side discussions, and then we tell them what they should be doing if they are going to help us for the balance on safety and specimen collection.
Thank you.
And any other questions? Okay, I have one. I'm quite curious how you encourage people to do all of these. You have great solution and techniques in these citizen science, but if the normal people want to do these, they have to be at least get some training. So how you involve them and how you usually train them to do all of this, because it's a bit complicated.
So what we did is we tried to make it as simple as possible. So we built a Facebook page to do community science. I know most of you here might be using iNaturalist, but iNaturalist lack a platform to do like direct answering and questioning, but Facebook platform provides this useful techniques, and you can also private message the collectors. So what we did is we built a Facebook public page, and then people can post what they found. If they are suspecting it's a bird collisions or not, they found a contact bird carcass, they post it, and then we will tell them what to do next. Like, you should have gloves, you should have a bag, and where to send them, how to send them, and what we are going to do them. And if they are curious or interested what will happen next after the necropsy, we will tell them that, okay, so this bird you found 2 months ago, if you're still interested, do you want to know what's the death cause? It's of bird collision because we find this, this, and this. If you want to solve the problem, what to do next. So this become to form a community. And then these ideas of how to prevent more deaths, and also to spread the knowledge of collecting carcass, because we know that most cases, people couldn't alter the changes so fast, but they can do some help in preserving this natural history data. They could spread these things, and then this is a community building up, like for now, like about 6,000 or 7,000 people joining in.
1,000, wow.
Yeah, so that means why we have a lot of carcass coming from around.
Okay,
Taiwan.
that's really cool. Okay, any question?
Thank you, such a great talk. It's not like a question, it's more like something I want to say. So can you guys come to Australia? We're in urgent of needing guys like you.
Sure, but we need funding because we are like self-funded to come to the conference. We need funds, then we can go to Australia.
Well, fine if anybody else would. Oh, myself, I don't have any money either. So thank you, though.
Okay, and it's time. And the next talk, still Chi-Heng's talk. She will talking about the individual. Let me check it. Sorry. Exploring individual story, rethinking specimen display in modern museums. Let's welcome Chi-Heng.
Okay, so I see someone coming in after the previous talk. So this talk is previously designed not to be given by me, but by another friend, but he decided not to come because he forgot the actual date of this conference. So that's why I'm here presenting for him. Okay, so then I'm going to do my self-introduction again. I'm Hsieh, and we've talked about the collaboration previously in the triangle between vets, specimen collectors, and citizens. And in the last part, I left the hook that we can do exhibitions showing related stuffs. And so this is one example which we have just done in the National Taiwan University. So in the past 6 years, I've been serving as a student assistant in the Museum of Zoology at National Taiwan University. If you Google this place, you will find that this is a university museum with histories. It was established in 1928 during the Japanese colonial period, and then people are collecting stuff, and it was said to be the home to more than 20,000 specimens. And there is an exhibition room located in the first floor for a life science building, though like most students who are studying life science, they have no idea there is an exhibition room. It's small, like about half the size of this room we are in, and it has been, though not a lot of students know it, but it has been a known tourist spot and attracted more than 10,000 visitors per year. So this may sound like a win-win that we have collections, we have places to exhibitions, but however, if you walk inside with such thoughts and thinking that you might have some really good experience, this is what you may saw. First, you will see there is like something written on the board in Chinese and like some English translation. This is saying that they are going to display specimens and talk about how animals produce sounds and without media. Yeah. And then you may see there is a trophy head of antelope, which is not a species found in Taiwan. And then there are a small group of bird mouse, skeletons of a cassowary and elephant, and some shells and logs in front of these specimens, which these are all part of the display left from the previous exhibitions. And this is like, we already know that the size is small, that's only half of this room. By disposing things left from the previous exhibition, this is just going to make the room squashier, and it's going to disrupt the logic flow of why these specimens are chosen and put there and arranged like this. To be honest, I will say this is just a bunch of mess, that you are disposing things and you are acting this like Arctic. So if we get closer, we can even see that the specimens are not like in good conditions because we are like university museums, we don't have conservators. And these specimens are left from the Japanese colonial period, so they are not really in a good conditions. And when we look at the labels, we see that they have only the names put on with no further information, including who collect them, all the field notes, where and why, all those kind of things. It's just empty. So this means that these specimens, which are placed in the exhibition room, are solely used as a display of the species for people to know that, okay, so I now know the name, and then the species might have an outlook like this, though we know that this is not in a good shape, so that you can just try to have a sense. And now, this could be replaced by higher quality photos and videos that we know that these taxidermies are not in a good shape. We can just find the videos and photos and show them, no, this is what the owl should look like. This is what the elephant should look like. And so then this comes to questions that we often heard visitors talking about. They are ugly specimens, and why don't we just, like in the traditional culture of Chinese is to let the dead rest, so rest in peace, and just don't do specimens, bury them, burn them, whatever, do some funerals. Why do we send them dead specimens? And this is also the same questions we are asking for ourselves, like as specimen preparators, why are we squeezing our time? We are students, we are squeezing our time to do this kind of things and dive into the preparation world. Therefore, this spring, when we are offered this opportunity to refresh this room, we had a group of students with different majors. So we come into a group, and some are majors on fish, some of us are major on birds, some on insects, and there's even a guy which I say he should be presenting here previously, that his major is really interesting. His major is actually on stage design and dress designs. So this means that he is not a person working in national history collections, but he has knowledge on national history collections, because what we all shared here for this group of students is we all have a passion for specimens and collections. And in some sort, to some extent, we are all specimen preparators, though we are not trained, mostly self-taught. So what we did is that we had this group, and from designs, including the drawings, all the layouts, and which should be where, and how the stages are designed, and also the woodcrafts. And this is fortunate because we have this guy who knew how to build stages. So we are using the technique. They build stages outside for outdoor stages for performance to display the specimens. And also the lighting, because he also works in gallery, so he knows how to adjust the lights to make the specimens to show better without shades. And we also changed the arts and labels. So like the guy previously presenting, talking about relay station, he actually has a good sense of drawing and art piece. So he's the one designing what are going to show on the labels, what are the drawings, we redo them and including labels because we want more information to be shown. And also to specimen preparation and selections. So it's all on us, a group of students, to decide which to present, how to present them, and what are the layouts, what the outlooks are going to be like. And this is like interesting and amazing because we never thought we could do this before and prove that, oh, this could be done with a budget limit about like, you can try to guess how much we spent on this exhibition. We spent like only 50,000 Taiwanese dollars. So that is around like 1,650 US dollars to do these exhibitions. So what we wanted to show here is that even though this is like a low budget exhibitions, we could still use specimens to tell various valuable untold stories which are not known and worth to tell, including what's the look inside the specimens. So people are really interested for visitors, what is inside the taxidermies, and like the carcass package sent from Citizen Science. So this also forms from the previous talk, like how we spread the ideas of how people should collect them and give them to museums and help this collection grows. And then to the seldom known collection histories we discovered through old newspapers, because we are finding that written words and there are like numbers of days on the newspapers, and we can tell more things on what is like only wrappers and how we made the measurements and more. So let's use this example. I should be having a something here, but no way, we can just do this. So let's take this young female pangolin as an example. It was found dead on June 5th last year, and it was found by a road in the suburban hills last year, and was posted on Facebook by someone who we don't know. So he is not one of our community, but we still see the post because someone reposted in our community. And it was one of our teammates who decided to go out and look for it and search for it. And fortunately, he found it. So therefore, we have an on-site photo of what it's looking like when he found it by the road on the suburban mountains. And after bringing it back to a preparation room, after washing out all the wriggling megas crawling around the scales, and we can look at necropsies because we have like some sense of how necropsies are done. And we found the multiple punctured wounds located on its lower backs, and also like the bitten tail tip with all the scales peeled away. And later, by dissecting the gut contents of this creature, we also found like there are 7 more species of ants which are known to favor to nest in different microhabitats, meaning that this pangolin in its last day has been going through different kind of habitats searching for food, and this is a record of species interactions. And through discussion, we later on decided to put it up as a flat skin specimen to expose the wound records, because we know that for taxidermies, it's hard to see the wounds because the wounds are not clear if it's in a full shape, and people might draw to the species. But for flat skin, they first start to think why it's like this, and then they might saw the wounds, and also by pinpoint out through like storytellings. And then we also display the skull to prove that it's not a roadkill, and also the studies specimens we sent to researchers, the gut contents, and also the notes describing the story I just told. So we hope visitors who came in and saw this specimen could know that this is not only a pangolin, but the pangolin, the young female pangolin whose last dinner was 7 kinds of ants and like some kind of vermin. She has been through all kinds of habitats, and unfortunately died because of feral dog attacks, which is a surging conservation issue for the species now in Taiwan, for all those pack of dogs running around and causing problems. An interesting thing we found is that by changing the way of telling stories with specimens, we are telling an individual story instead of just simply showing the pangolin and telling that, oh, so feral dogs attacks are a problem for pangolins. We are telling her story. We have been having sympathetic visitors doing recreations. So like here we've shown, this is an example of a comic who visited our exhibitions and then decided to draw the story out. And this story has spread on Facebook and all other kinds of media, meaning that the story or information in the environmental education wanted to spread get further into different groups of audience. Now that this exhibition is like 95% ready, still adding things and adjusting to make it even better. For example, here we've been hearing people to put up photos or images or like different kind of cars or like different kind of things. We can add them later when we go back. We did several trials, including trying to display every form of specimens we know to show diversity of possibility, including wet specimens and dry specimens, from taxidermy to study skins, all the way to different sample parts. We've also decided to present the display in the term of diorama, which is the common way of displaying specimens in the common world, or like a traditional display way, to let visitors to feel the difference in between the show of modern display in a diorama most of the times, and then the way of displaying an individual story. What do they feel? And if they are really interested in the individual stories of those placed in the diorama, they can still find the stories with a QR code placed inside the room. Therefore, we decided to give this exhibition a theme name, calling the double dissect. So we are playing a little bit with the word, for there are 2 different layers of dissect. The first layer of dissect is on how specimens are prepared, and then the second layer of dissect is on the thoughts of the preparators. So this is like trying to open up what's going on our minds and our discussions behind the specimens. So lastly, let me do some advertising, is that if you are ever going to make a visit to Taiwan, it's like another 1.5-hour flight from here, Okinawa, to get to Taiwan, Taipei. We are welcome to have your visits and hear your thoughts and opinions and comments. Because we are in charge of curating this, we can always add something to it. If we feel this is a good idea, we can just always put something on it. And it's free to access, meaning you don't have to pay the ticket. And if you would like to have us to go through the exhibition with you and have further discussions to talk more about something that is not displayed, but we know all the stories, because every specimen placed in this exhibition, about 150 specimens, are all made by us, the group of students. We know the stories behind them. So it's welcome to find us on Facebook. And though the advertising is written in Chinese, you can always translate with like Google Translation or the Facebook translation app. And like, you can always find us, and we can have the students, because we are at school, the student collectors can come down and we can have a special book trip tours. Thank you.
Thanks, Hsieh-Heng's great presentation. Yeah, and any comment and question?
So Elie Mario Saliba from Paris Museum and Sorbonne Université. Because I work between 2 organizations, a university and a museum, I have to say that the university also has a collection. I am absolutely amazed by what you did, because our collection at the university, there's only 1 person working on it, and 1 person on zoology, 1 person on botany. They spend so much time to be able to do something, because it's also a teaching collection, so you don't have any valuable information for natural history. I just wanted to ask you, how much time does this kind of work take? Because it's amazing what you're doing, and how much time do you need to do this kind of thing?
Okay, so we have this idea of arranging exhibitions someday when we're preparing the specimen. We notice the seeing the exhibition room is not in a good state. And for putting up these exhibitions, it spent us like half a year. So it starts from like spring, like around April, we have this discussion, we write a paper form to give to the professors to say we wanted to renew this, and this is what we're going to do. We wanted to talk about the stories of individuals, and then we prepare the specimens like in a rush time, these 2 months, conservating if some things need to be adjusted and fixed a little bit, and then we put this up. And because we have like students who could help us all around Taiwan, we even have our wood decks. Actually, they are done in Taichung. This is the middle part of Taiwan, and we added some basic wood crafts, and then we move them inside and to do more. And the whole thing was put up using, I remember like a week, right? Yeah, a week. So it's just like to prove that sometimes you can just encourage students to join in. Students might have some ideas and they are free labors. That is why we can do it like for around $1,600 US, because we can't pay on ourselves and we are willing to do this. Students are really enjoying to get engaged to show that their thoughts are being expressed and known for people.
Okay, and there's a question from the internet. What kind of difficulties did you meet during this process?
Okay, so the biggest problem is that we have to communicate with the staff members who is the collection manager of the previous exhibitions, because like we know that the previous exhibition is not in good state, but we are students, so the identity is not like to match to like challenge him that you are not doing a good stuff. So we have to like alter the words and try to sprinkle around to tell him that we are trying to make things better together, and then put this up and try to let him to, not to cause too much problem, but just stay aside and we will do everything for you.
Thanks, Chi-Heng's presentation, and it's time, so we have to go to the next talks. Thank you. Next talk, is from Taiwan, Jing-Fu Tsai Jing-Fu. He's an insect guy and from the National Museum of Natural Science. He's also one of the leader of the collection and the research. Please welcome him.
Thank you. Today's the following 2 talks is all about the type specimens and how to use them well to classify the taxonomy problems. So this kind of animal we call the jumping plant bugs or jumping true bugs, the Isometopinae. And actually, it's a very highly unique, as you can see, possessing the pair of ocelli on the top of head, which are absent in all other members of the plant bug family Miridae. So you can see it's very tiny. The body length is about 2 and 3 millimeters. And they are just living on the bark and crevice. And but we know very little about their bionomics. So we only can see that they have a very big eyes, and you can see that the head is strongly anterior posterior flattened head, but they do have some jumping high leg, and also frequently intercept by the interception flight trap, like mice trap, indicating they might have some flight ability, but they are really rare in the collection and the poor bionomics. We don't know about their habits and the food and reproductive behavior. And but I saw some papers that mentioned that they are feeding on the arthropod, but this is only for the artificial rearing condition in the lab. But as when I doing the field observation, I can see they are probing. They can just searching and probing using the piercing, sucking mouth parts to just searching some food, and maybe some literature said it can just feed on the epiphyte growing on the bark, such as the lichen. And they are frequently occurred on some specific trees like Himalaya ash, Chinese banyan, and tung oil trees and maples. So as you can see, if we get close, look on their head morphology, you can see they have a very big eyes, and top of head present two little eyes we call ocelli, and head is strongly, as I mentioned, anterior posterior flattened. And you can see some of the group have a very enlarged abnormal antenna, and sometimes they have horns here. And let's check the videos. It's very interesting. You can see how the antenna function, and they're just keeping waving and on the bark and touch on the bark and searching something. And during working, and they are just, the mouth part is always probing on the bark. So I think it's very likely to search some food. And the larvae, as you can see, it's very diverse, very weird. And they show striking diverse morphology. Some species are very flattened, and some others are easily recognized, bearing with horn antenna. So they are quite cryptic and concealing on the bark. So in the world, there are nearly 290 species worldwide, and about 80% represents in the old world, extremely diverse in Alpha tropical and Indo-Malaya regions. So they are actually thermophilous, so they just occur in tropical, subtropical, humid, continental climates in temperature zones. And based on the phylogenetic trees, they are quite basal here, quite basal lineage, ancient lineage, and sister to the rest subfamilies of Miridae. So when we examine the types and the vulture specimens of 17 species in our museum, because we have many types here, and surprisingly, only a single sex have been documented for several species, which show in the blue text here. These are, the blue in text are only one single sex. They are all types. And so I have a chance that, in Taiwan, the citizen scientists are very active in the social media. So they post lots of interesting, vivid photos from the fields. And this attracted me to just contempt them, and they lead me to just discover several of them are the same species with very strongly sexual dimorphism. For example, for these two species described in the same year, overlapping period, and same locality, as you can see the label here. And I can see this by punctata only female exists. And the other one in Hsieh is male only. So as I compare these two, and I just think over that they might, except for spot patterns, I cannot tell any difference because, for example, the head morphology for me is the same. And in the field, we just do some observation, and we found that these two types of forms, they are actually sympatric, and they occur on the stand tree. So you can see they frequently occurred on the Himalaya ash and the tong oil trees. So so we believe these two are the same species, they're just the sexual dimorphism. Yeah. And the others, another case is these two species. This one is described from Taiwan, and the other, but this is only the species, the type species are male, and the others is described from Yunnan in China, and as we can see that these two are really different. But during the field work, we found these two forms occur again. They are sympatric and occur on the stand trees. It's banya trees. and just two different sexes. So again, these two are the same species. So through the information from the citizen scientists, this is the third case, and these two species were described from the same locality again, and the same date, period. And you can see the male has a long wings and is under the name of the Sophians formosanus. And the others is the females with very short wings, and it describes another species. So this is again they are just the conspecific and the same species, but with the different forms. So so this is the case that we found that some species have a very strong sexual dimorphism, and they can co-occur together. And based on the field work, then we can just make very sure that yeah, they are the same. And we do have a extra new records and species not well studied, and they are just waiting for the examinations. And this is the one that they just think this is the misidentified, but currently they don't. The previous authors think these species should be these species, but as you can see, the holotype and the real specimens are quite different. So this is maybe undescribed. And luckily, we have both sexes, so we can just make examination in the future. So so as you can see, in Taiwan, we harbor a lot of species, and it's quite remarkable. We are for the jumping plant box, we have about 20 species, so including undescribed. And you can check the neighboring regions. Still, the current number of species are relatively low, but I think it's just not in well study. But as you can see, such a small island, we have such abundant species. It's just very cool and amazing. So the future taxonomic work on this group, I think is required the use of diagnostic characters. The diagnostic characters that should be gender neutral characters, and we have to use, in some cases, we have to use the molecular identification, and also we have to go to the field and just observing their life history to overcome the current taxonomy challenges. So enhancing our understanding about this, about their diversity. So I really thank those citizen scientists that they provide me very valuable information and very good photos, and let me just go to the locality and check whether they are really co-occur and what they are doing on the bark. So I really appreciate that. So thanks for your listening. So we have about 1.5 minutes or 1 minute. Okay.
Any question or comments? No? No? Then I would like to say that it seems citizen science is really prosper in Taiwan.
Yeah,
right?
actually, yes, citizen science is very active in Taiwan, because in Facebook, we have all kinds of groups, insect groups, and they are very specialized. Even we have some Socoptian and many kinds of insect family groups. They just form a club and keep posting a lot of photos on, and we can just communicate with them and to search more details about the insects. Yeah.
And any questions and comments? No? If no, then next one still Jing-Fu, right?
Yeah,
Yes.
that's why it's me.
He's going to talk about a century-old taxonomy problems in jewel box. Okay, welcome.
Yeah, again, this is about the type specimen study. This is resolving the century-old taxonomic problems in jewel box of the genus Scutellera. And this jewel box is a well-known family of worldwide. It's about 500 described species distributed in all continents. And as you can see, the important features of them is that the Scutellera. The Scutellera is always enlarged and conceal the wings and abdomen. So that's why, because they are very bright, so that's why in English they are frequently called jewel bugs. But you can see many of them are not so bright, they are just dark brown or black. So the type genus of this family is Scutellera, which is the oldest genus of the family, in the family. So it was established by the Lamarck, who only praised one species in the genus, the Cimex nobilis, described by Linné, and subsequently cited by Fabricius and Sousa. He only provided a very brief and unclear definition of this genus, emphasized the enlarged Scutellera. So in the first half of the 19th century, the genus Scutellera was understood in a broad sense, and early authors praised many different kinds of pentatomids, processing and enlarged Scutellera into this genus. So as you can see, the Scutellera in this broad sense was a very heterogeneous taxa, containing highly disparate forms. Some of the species earlier praised into this genus even did not belong to this family. For example, the Pentatomidae is a Prataspidae. So even some other pentatomids, they are praised in the wrong genus. So it can be, so this, for example, this now is belong to the genus Cantal, and this belong to another genus, and this one too. So how to define the Scutellera in the modern sense? For this, we should first know what the type species of the genus is. So we may remember that the Lamarck original including one single species in this genus, so Cimex nobilis described by Linné, later cited by the Fabricius and the Sousers. So what is those species? So we have to first to check the Linné Cimex nobilis in the left here. And actually it's not the Scutellera is transferred to the Caulifera now. And the other three, all the authors, including Fabricius and the following subsequent authors, Sousa, Stoll, and Schroeter, they all misidentify the Linné's species. But the Western Wood correctly, the first three correctly recognize the Fabricius misidentification. So in his catalog of Hope's collection of Hemiptera, he lists the broad form of nobilis of Linné is not Fabricius. It's clearly pointed out. So the narrow form is a different, is an unnamed species. So he proposed the name Tactocoris perplexa for it. So he clearly pointed out that is the same as the nobilis as recognized by Fabricius, but different from nobilis of Linné. So he resolved the misidentification and properly proposed a name for the narrow species. So this species becomes the type species of the genus. So let's see how the species placed in the narrow defined genus. This is the first one, the one which we discussed above, Scutellera proposed by the Western wood for the species misidentified by Fabricius, the type species of Scutellera. So where is the type now? Unfortunately, because it's too old, and in the old time they don't put the type labels on, so nobody knows where's the type. Maybe type is still there, or maybe type is still or lost. So it's not, we just cannot find. But the identity is still very clear, because I think the old ancient book, the illustration is also clear enough to identify them. So we did not designate a nail type for this. And this is a very important pest in India. And the host plant is a Jatropha plant. And the second species, this is the second species of the Scutellera. And as you can see, it's quite different according to their black marking. And you can see the black marking a little bit resemble a Chinese script called rice. And yeah, so the name of this species is perhaps even more confused than the previous species. It was first named by the Lichtenstein in 1796 as Timex amicitinus, but that means it's purple. It looks like purple. But in a work which was later suppressed by the ICZ, and therefore it's become an unavailable name. But later, 2 years later, in 1798, Painter proposed another name for this species called Timex fasciatus. But this name is a junior secondary homonym of another animal, this one. This is belong to another family. So it's become the homonym, so it's become invalid. So it cannot be used. So in spite of this species still very frequently cited as Scutellera fasciata. Again, primarily, it's just wrong cited, yeah. So so finally, the Western wood, he proposed this name, the first one he proposed. So this is the oldest name for this species. And subsequently, this species described again and again for 3 times. So if we check the type, and we can just correct the name. So the valid name, of course, is by Western wood. So all of the subsequent names are just become synonymized. And very interesting that these old species from the Asian continent, they always show the green background. The black marking is not so variable, but you can see there are 2 different colors. And we cannot find any transition form from the collection, the green one and purple one. So we propose 2 subspecies for Asian mainland and Malay archipelago. And although they looks very similar, but still the genitalia, both female and male, are very different. Yeah. And the third species is from India. This species is also quite not very young. It's relatively old, and it is very rare in the collection. And they have for 2 names. So the valid name is the valid name is Spirogastra, described by Walker, and they just occurred. Their distribution is very restricted, just South India and Sri Lanka. And we don't know about their anything. We don't know the host plants. We just documented from the collections. And the last one is this species are from Java, and the type is in the Polish Academic of Sciences. We have a chance to to go there and re-examine the type. And we finally think this is not this genus. This belongs to the Africa genus. So the Java may be wrong. I mean, the locality may be mislabeled. So we just remove this genus, this species from another genus. So it's not belong to this genus, the Scutellera. So as you can see, such a small genus and loaded with so many taxonomy problems, so many misidentification after the name, and so many new synonyms. So I would like to thank, because we travel many museums, maybe more 20, so we thank all the curators, the collection managers, assistants to clarify these problems. And thanks for your listening again. Yeah.
Okay, any questions and comments? We have 5 minutes.
2.
Okay, 2 minutes.
2.
Okay, whatever. I have one question. I'm not insect guy. I don't know the system and the taxonomy, but I'm interested about the color. And it seems they have so many color and very obvious. Any function of these colors?
Yes, they are very colorful. And so far, there's no study to study what is the function, but they are just like beetles, because most of them have a metallic, the nanostructure to refract. This is a physical refraction, but even it is physical refraction, still some of the green metallic jewel bugs, when they die, they will from green color to blue color. And so I read some papers that they are very, you can see it's very bright, so you can easy to spot, you can easy to see them. But you can imagine if this box, the shining metallic blue or green colors, no matter it's beetle or heteroptera, when they are in the forest and they are sitting on the background of leaves under the sunshine, and you can see they are just mimic in the forest. So in the forest, you cannot easily to spot them because they are just merged into the background because of this, yeah. And for those very dull brown or yellow, they are not easy to find, because if you want to collect those beetles, because they have some specific host plant preference, you only can know that their host plant, then you can collect them. So impossible to collect them if you know nothing about their host plants. Yeah. So that's, I mean, you mentioned about the color.
The color, yeah. So at least part of the function is about camouflage, right?
Yeah.
Hiding in the habitat.
Hiding. Yes.
So no function for species recognition or sexual selection? No.
They do have some. I mean, very interesting that one of the shining jewel box green one, they have some, because the marking are very characteristic. This is also for the sexual recognition. So the female and the male, some of them they can make a dancing. Before mating, the male have to do some dancing behavior, and the female, if the dance is not perfect, then the female may reject. I got some videos, and yeah, this is really cool. Yeah.
Any questions and comments or suggestion?
Yeah, but anyway, I mentioned these two topics, that study type is very important, because if we want to get the clear identity, then we have to, do a lot of taxonomy work. I mean, describing new species is very easy, but
One comment. So you visit so many museums,
yeah, I visit about 20.
20 for the
Yeah.
all of the
Yeah, all of
museums.
this, all of
So-
this. But so this topic start from 2016.
Yeah, and so you show for the type photograph of the types. So here for the spinach, and do you have any idea if you don't go to the museum, but you got it take for the picture, so you are for studies more easily to progress.
Yeah,
Yeah,
I
and
think so.
So
I
you have
think-
some idea of the. You don't spend money for traveling, but you got it for nice pictures. So it's a good way for the.
Yeah, I think this, if the museum can provide the digitalized images, then, yeah, sorry. Yeah.
So I actually manage a collection of fossil insects, so not modern insects, and we actually do this quite a bit. So I actually work with some grad students in France, and they told me not to mail the specimens. They just want me to take the images, and we'll email back and forth. And so with the time difference, I'll take a picture, I'll email it, they look at it the next morning, they look at it and say, oh, can you take a close-up of the leg or this part of the wing? And we just go back and forth like that and we call it a digital loan. And we go back and forth like that because we want to track use of our collection. And it's been really great. So we have a lot of specimen images like on GBIF that we've only identified to maybe family level if it's good, because they're, insects, there's a lot of them, and fossil insects, there's a lot of new taxa. And they'll just go look through our GBIF photos, and then they tell somebody else in their lab, oh, you're working on this group of insects, go look at Colorado's insect photos on GBIF. And we've had so many new taxa described from our collections just from images like that. It's been really amazing. And these graduate students are never gonna come to Colorado to look at my collection, but all my images are on GBIF, so they can just sit in their lab and do it from their office. It's amazing.
Yeah, so I do hope that through such organizations, Spinach and TDWG, if the museum can just upload they maybe not too much efforts to take photo in very detailed character, just the image of the habitus photos. That's maybe enough for the professional taxonomists. They can easily to recognize this is their identity. So I think this is very important that if the type specimens photo can be, searchable from the
Comment. So I came from the Hokkaido University Museums. So we have for a primary type almost 10,000 primary type we have, but we are not sure which is a priority. So if for some project, so you propose the project of this genus, so the curator or collection manager take pictures of more first priority. That is more, one of the project. Yeah.
Yes.
Just a comment.
Even digitalizing the type specimens also need to some strategy. But for big museum, they have thousands of type specimens. But so it's a big work too. Yeah. But a simple image is enough. I mean, it's enough. So if it is accessible, and if the taxonomy want more details, then he can just contact the curator and just show them the photo. I need this view of close-up photos of the characters. That is also possible to accelerate the problem of the taxonomic issue. Yeah. Okay, thank you.
Okay, the final talk, Yu-Hsiang Ho. He was in our museum and now is in University of Sydney, and he's going to tell you the story about biodiversity of the other beetle story in Taiwan. Okay.
Okay, thank you. Hello, everyone. My name is Yu-Hsiang Ho, and I used to be the research assistant in the National Museum of Natural Science in Taiwan, and right now I'm studying my PhD degree in the University of Sydney. So today I'm going to talk about the biodiversity of myrmecophilous histeridae in Taiwan, which is a small project I used to study in Taiwan. So in yesterday's lightning talk, we've heard that some researchers found new species in museum collections. And in this kind of case, it's quite common in entomology studies because many groups are poorly studied. And the presentation I'm going to talk about today is this kind of case. So Histeridae. Histeridae is a moderately diverse family within Scutellera, also known as beetles. There are 9 extant subfamilies within Histeridae, and among them, 2 of them, Chlamydopsinae and Heterinae, are known as all species live inside a nest of social insects, and mainly ants and termites. Chlamydopsinae is a relatively small subfamily within Histeridae. There are around 13 genera and 179 species. Most of species of this subfamily distributed in Australia. And they also distributed in Oriental region and Palearctic region, with northmost to Japan and westmost to India and eastmost to Fiji. The host records of this subfamily is also very rare, with only 37 species being recorded. And the host species scattered in 4 subfamilies of ants, including Ponarinae, Dolichoderinae, Formicinae, and Myrmecinae. Compared to Chlamydopsinae, Heterinae is more diverse and well-studied, with around 111 genera and around 335 species. This subfamily mainly distributed in Neotropical region and Nearctic region, with several species distributing in Palearctic region. And the most well-known host of this subfamily is army ant of Dorylinae. Some Heterinae even have elongated legs and will migrate with army ant. Besides Dorylinae, Myrmecinae, Formicinae, and Ponarinae are also the host of this subfamily. So in Taiwan, currently we have 1 species of Chlamydopsinae and one species of Heterinae. And Taiwan is also Taiwan is also one of the locations that have both subfamilies distributed. Another one is Japan. And Eucaryotiopsis mirabilis is the only species of Chlamydopsinae in Taiwan. It's endemic and also the type species of this genus. Heterus optatus is the only species of Heterinae in Taiwan. It was originally described in Japan and also in Korea and was recorded by Mazu in 2007. However, through my extensive collection in the field and also examination of specimens in multiple institutions, I found out that the Taiwanese myrmecophilous histeridae, the biodiversity is highly underestimated. It's like, so the aim of my study is trying to explore the diversity of Chlamydopsinae and Heterinae in Taiwan. Also, I will revise the host records of these beetles, which is totally unknown to Taiwanese fauna before. So this is the revised checklist of myrmecophilous histeridae in Taiwan. Originally, there's only 2 genera and 2 species. But right now, there's 3 genera and 7 species, with 5 new species and 1 new record genus. The first one I'm going to talk about is Eucaryotiopsis. This genus is right now the most diverse myrmecophilous histeridae genus in Taiwan. Have 4 species, with 1 is on the left-hand side is Myrmecophilous mirabilis, which is the already known species. And the rest 3 are all new species. And I'm going to name them SP1, SP2, and SP3 in my following presentation. Eucardiopsis mirabilis widely distributed in entire Taiwan island and mainly found in mid-altitude forest. SP1 and SP2 are only known from one location in central Taiwan, and they are symmetric with mirabilis at least at that location. And SP3 are mainly distributed in southeastern Taiwan and symmetric with mirabilis. The new record genus of my study is Orthoceles. This genus have 2 new species in Taiwan. Their distribution in Taiwan is relatively restricted compared to Eucardiopsis, with 1 species in Miridae Island, which is 33 kilometers from eastern coast of Taiwan. And another one is only known from 2 locations in southeast Taiwan, and they mainly distributed in lowland forest. Orthoceles currently have 18 species, with 10 species in Australia, 1 in Papua New Guinea, 6 in Indonesia, and 1 in Ryukyu Island. So the finding of this genus in Taiwan is actually not a huge surprise, since there's a large distribution gap between Ryukyu Island and Indonesia. The species of Orthoceles on Ryukyu Island is Orthoceles Hsieh. This species was firstly described in Yonaguni-jima Island. Although the Taiwanese population is very similar to the Ryukyu Island species, but currently I still treated them as different species, since the shape of the pronotum and the shape of the humeral depression are very different. And with the finding of this genus in Taiwan, we partly filled in the large distribution gap of this genus in Southeast Asia. And we can also expect that maybe the Philippines and Borneo have this genus, but this may need more study and more field survey to find out. And the final one is the Heterinae. I didn't find any new species or new genus within Heterinae. So there's only 1 species, Heteris aptatus. This species is widely distributed in central and south Taiwan and mainly distributed in low to mid-altitude forest. During my field survey, I've managed to find some histerids inside the ant nest, and that is a good chance for me to record the host species of these beetles. So in my study, I find 2 species of Eucardiopsis and Heteris aptatus inside the ant nest. And the host of both Eucardiopsis species, Eucardiopsis mirabilis and SP1, are of Phytoephorbiida, which is a widely spread ant species in East Asia. And the host of Heteris aptatus in Taiwan is Aphenogaster tipuna, which is very interesting that because the host species of Heteris aptatus in Japan is another species of Aphenogaster. And besides record the host species, I also managed to record some video of the interaction between Histeridae and the ants. So most of the myrmecophilous Histeridae are considered to be symphiles, which means that the ant will guard and feed those Histeridae beetles. For example, the photo showing that this is a species of Heterinae described 7 years ago, and it is well known for it will bite on the abdomen of ant and carry around by ants. Besides symphiles, a small portion of Histeridae are considered to be predators. They tend to have larger body size and will prey on like workers or larvae of ants. And I managed to find to obtain 2 species of Histeridae with ants. One is Heteris aptatus, another one is Eucardiopsis SP1. All of my Heteris aptatus specimens with ants are collected in Hui-Sun Forest Reserve in central Taiwan. I found them under the rock in the nest of ants. And I record some of the interaction, and we can see that heteros updated inside the nest. First, I found them are being the scavenger inside the nest because they will search around and looking for anything they can eat, and mainly the corpse of other insects. Also, the ant will try to feed the beetles as shown in the video. So it feels like the ant consider the beetle as their larva because besides trying to feed in the beetles, I also seen that ant, the worker trying to carry beetle around. And I also found that in this video, the beetles are grabbed on the larva of the ant, and it seems like it's trying to update the chemical odor of the larva, and maybe that is the like chemical camouflage, so that the worker will consider the beetles as their larva. But this may still more experiment to confirm that. And another species is Eucardiopsis sp1. This species is collected in Beidongyan Shan, also in central Taiwan. Because this species is very rare, so I only record a very short video. But in this video, you can still see that the worker just ignore the beetle. So it may have the same like chemical camouflage like the heteros updaters. Yeah. So this is my presentation today. I would like to thank to Dr. Hsieh Nomura and Professor Masahiro Ohara, which is on the stage, for loaning the specimens, and Dr. Fengquan Xu for helping me identifying the host species. Also, Dr. Wen Liang, Mr. Renhao Liu, and Mr. Binghong Ho for providing numerous specimens to me. Thank you very much.
Okay. Thank you, Yu-Hsiang. Dr. Ohara, do you have comments? we are a little bit delayed, but I think we can have one fast question.
Yeah, Eucardiopsis is a very rare beetles, and I never see for such video. It's a very impressed. Thank you very much. Thank you.
So I think time is up. Thank you again. And I think this is the end of this symposium. And thanks for all the speakers and your attendance this symposium. Thank you.